Welcome to you all, and I think it's indeed a great pleasure to have with us the most eminent speaker, Dr. Srinivas. Similarly, we have with us our director from Sri Vishnu College of Pharmacy, which is our sister concern college at Bhimvaram. And I would like to thank all the people here, majorly the uh, all the participants from across the India, who are around more than 1,000 participants and with 28 states and I think 3 countries covering abroad. And I think it's indeed a wonderful event being organized by the convener, Dr. Vanita, and the main action plan was done by Bhavani. Thank you so much for organizing such a wonderful program. And it's an 1 week online international FDP program starting from today till 11th of July. And I'm very happy that the topic is on the current research trends, and I wish that we as Vishnu brand is very popular, and I wish that such programs can definitely deliver better tasks in future. And being the only college in the state of Telangana to have both NBA and NAC, and once earlier we also got NIRF, and I think we constantly get upgraded with the current knowledge. And I think I'm very thankful to the society for creating a wonderful atmosphere and an ecosystem which is very congenial for the development and growth of the staff as well as students. Above all, I would like to thank all the resource persons for having a wonderful time in the next couple of days, and I wish that this program is going to be a successful one. And above all, last but not the least, I would like to thank the backbone of the society, Sri K.V. Vishnuraju Garu, similarly the Vice Chairman, Ravichandran Rajgopal, and the entire team of the society for staying as a backbone in running a quality education across the society. Thank you one and all. With this brief introduction, I hand it over to Dr. Vanita, who will introduce the speaker. Thank you.
Thank you, sir. Respected Principal Dr. Ramesh and our Director Dr. Kumar and all the participants, a very good morning. Today, it's my honor to introduce the eminent speaker, Dr. Srinivas Bullapalli. He is an expert consultant in drug discovery, pharmacology patent at Expert Pharmacology Consultant, Mumbai, Maharashtra.
Thank you.
Vanita, your voice is not audible.
Now, sir?
Yeah, now it's fine.
He is a passionate and determined scientist who brings a great degree of competitiveness to the work environment. He is highly motivated and very determined researcher who takes up any challenges and not afraid of thinking out of the box to accommodate novel ideas. Needless to say, his multiple patents and research publications in international reputed journals is a good achiever. It is a pleasure to have his acquaintance under the academic and industrial settings at both professional and personal levels. He has completed his PhD from NIPER Mohali, B.Pharm and M.Pharm from Andhra University. He has worked in many industries under this M2 Pharmaceuticals Limited, Glenmark Pharmaceuticals, and Dr. Reddy's also. He worked as a postdoctoral scientist at Mount Sinai Medical Center, New York, USA. He has expertise in drug discovery. He has more than 14 years of experience, and he has a credit 8 product patents and 6 method of use patents and 18 international publications. So with this brief introduction, I welcome you, sir, for this conference. I request to enlighten our participants with your expertise. Thank you, sir.
Thank you very much for kind introduction, Dr. Vanita, and very also Ramesh and Dr. Bhavani and Dr. Kumar for facilitating this, and a good introduction. I could get to know more about Vishnu Institute of Pharmaceutical Education and Research. Thank you very much for the introduction. Dr. Vanita, may I have the
Yes, sir.
presentation? You can ask your IT professionals to post.
Yes.
Especially in present times, a lot of things, these webinars and all, like, for everybody, we all need to, thanks to the technology that is available, which actually facilitated all of us to have this kind of knowledge sharing and educational and other knowledge sharing and research experiences. Without the technology, we cannot even think of foreseeing such kind of sessions. In any case, today, am I audible to everybody?
Yes, sir. Yes, sir.
Okay, thank you. Yeah. So, thank you very much once again for inviting me for the series of webinar series of faculty development and student development program of your esteemed institution. Today, my topic is going to be I'm going to give a broader preclinical perspective about the process of Yeah. So before going further into the seminar, I would like to let us see the definition. What the definition for drug substance? A drug substance or a medicinal substance can be generally defined as any agent which can have a potential use in the treatment, cure, prevention, amelioration of a disease or disorder. It is for any process, it is important to define what is cause and effect relationship. In this, obviously, the disease, human disease, is the cause or the problem which we have to address. And the most, as we all know, the disease is biological in nature. So normally in drug discovery process, pharmacology and biology, pharmacology and broader biology is considered as the heart of drug discovery process. And here in this particular aspect, even though a drug substance can be a novel chemical agent, a small molecule, chemical molecule, or it can be a biologic like an antibody or a vaccine. But I would like to restrict my talk to the novel chemical entity NCE based small molecule research. So here, obviously, drug substance is going to be a medicinal product, like a medicinal chemistry, small molecule chemistry product. So obviously, in the drug discovery process, medicinal chemistry is going to be the brain behind providing the solution to the problem that is associated with the biology, that is the disease. As you can see in this slide very clearly, that drug discovery is a highly complicated process involving various expertise and experts, and that too experts requiring a broader interfacial understanding. So this is very essential for a successful drug discovery program. Lot of people, lot of people in the art as well as the commoners, they regularly ask, oh, is it really so complicated? Is it kind of a rocket science or what? This kind of colloquial questions we normally come across in the field. What I would like to mention is, yes, drug discovery research is really very complicated. It is much more complicated than rocket science. The best example is we as a country, India, we have launched, our country has launched many rockets and satellites, but in the drug discovery process, research, we are lagging way behind. So that gives a very good indication. This is just to highlight the importance of drug discovery and also the complexity that is associated with drug discovery. As you can see in the slide, normally, probably what I feel, and every pharmaceutical research institute and pharmaceutical students, everybody should appreciate one thing, that at the outset, pharma students, pharmaceutical graduates are the best people who are having a broad interfacial experience of various aspects that are taught to them in their graduation and master's programs. So normally there is an infamous saying that pharma students are jack of all trades. So with that, if students inculcate the habit of attaining expertise in so that then that will make the pharma students the best researchers that can fit into a successful drug discovery program. The next slide, I just would like to highlight the process of drug discovery and the various convergence of convergence of various disciplines and experts that are required in a typical drug discovery process. As you can see, you may need an information technology people for the data searching, and you need a intellectual property and patent experts, and you need medicinal chemists, you need protein chemists, biophysicists, protein biophysicists, and for modeling purpose, and chemists, and in general various kinds of pharmacology and broader biology experts like pharmacologists, toxicologists, immunologists, biochemists, molecular biologists. So it's a confluence of various expertise. So that coming together, which also highlights how complex the discovery process is. Coming to the next slide, various stages of drug discovery and developmental process. One can easily understand.
Slide has not moved. Sorry to disturb. Slide has not moved. Now it's there. Yeah. Okay.
Slide
go
is
ahead.
not moving.
Yeah. Go ahead, please. All right.
Is it right now?
Yeah. All right. Go ahead.
Are you guys able to see the slide?
Yes.
Okay. Yeah. So if we see the different stages of drug discovery, the entire when we take up a drug discovery program from lab stage to to the stage of approved product, there are several stages, as you can see, where verification stage and pharmacology, toxicology, and pharmacokinetics and drug metabolic clinical stage. And then after that, there will be a clinical stage of development, which is a major chunk of time and resources it takes. So, as a minimum of anywhere 12. Uh, am I audible?
Yeah, please, sir. Go ahead, sir, please. We are able to hear you, sir. Please go ahead.
It's fine. I think there is in between there is some connection problem. I am not able to. I am also not able to see the slide.
Okay. I think you have to take the screen, sir, and you need to control it.
Yeah. Is it fine now?
Yes, sir.
Yeah.
Yes, sir.
So.
Please. Please continue, sir. Your video is
Yeah.
off.
Yeah. So the whole drug discovery process may take anywhere between 12 years of time and $1 billion US.
ఆయన చేతిలోకి వెళ్ళలేదు ఇప్పుడు. Please continue, sir. Hello.
Am I audible now?
Yes, sir. Yes, sir. Please go ahead, sir.
Okay, it looks like there is some issue with the slides. In any case, I remember my slides. If I am audible, I'll go ahead with the slide presentation. You people just stream the slides.
Sure, sir, please.
Yeah.
I hope these all are the initial technology problems. Yes, sir. Please.
Yeah. Can I go to the if somebody can access is accessing the slides? Can I go to the slide number 6, please?
Okay, sir. Please go ahead. Are you able to see the sixth slide?
Yeah, that's what I think. There is some issue with streaming. You are able to see me?
Yes, sir.
Okay.
You can take the control and change the slides like earlier you did it.
Yeah. I'm just trying to understand. Um,
We'll continue, sir. Anyway, we can help you out.
okay. Can I go to the slide number 6?
Slide number 6. Yes, sir. It is already there. I think once again we'll access it, sir. Please.
Yeah. So basically as you all can see, the drug discovery process, as I explained, the clinical stage of development takes a major 70% stage, whereas the preclinical stage constitutes around 30% of the time and resources. And in the next slide, slide number 7, as you can see, this slide probably tells about the difficulty and just in the case of one particular drug to get approved in a particular drug discovery program, we may have to synthesize several thousands of compounds and put them through screening procedures at various stages of drug discovery to ultimately achieve one particular approved product. And at the end of the day, it may not be. Sometimes we may not be lucky enough to have one drug product also. So that entails the complexity in drug discovery. And one more aspect, regulatory authorities, being FDA or European Medical Agency, this kind of regulatory authorities also, from time to time, they raise the bar for safety of the drugs. So that altogether also complicates the whole drug discovery and developmental process. In the next slide, in the next pictorial, I would like to give an overview of various things that normally happen in the drug discovery development. Starting from target selection, validation, and where you need to have an excellent understanding of the disease mechanisms, pathophysiology, and then with the help of cellular and molecular studies to identify which particular drug targets are druggable, and then which are relevant for the disease area, what we are looking for, and then having selected a particular drug target, and one need to do and several studies to validate in vitro validation studies, as well as in vitro as well as in vivo validation techniques. And then coming to a discovery phase, where having selected a particular drug target, the pharmacologists and biologists have a have to develop various kinds of in vitro screening assays in order to have various screening assays so as to develop methodologies to screen compounds, and medicinal chemists, colleagues from medicinal chemistry, they start synthesizing various hit molecules. They start synthesizing based on initial knowledge and compound library screening to arrive at a particular hit molecules, and then based on that initial hit molecules what they have identified which are relevant for the drug target, then through the SAR studies and all, one need to further develop it, and to have several lead molecules are from different. It is important. So then the next stage is normally a lead optimization, what we call lead optimization, where a particular lead pharmacophore is optimized for better druggable properties with the use of putting them through various screens, in vitro screens, and then have a final lead molecule in order to have a drug candidate, preclinical drug candidate for extensive studies in pharmacology, efficacy pharmacological studies, and safety pharmacology and toxicology studies. So in the next slide, what is the importance of choosing a strategic disease therapy area? This is one of the most crucial aspect in can I go to next slide, please? I have crossed a few slides. So choosing a strategic disease therapy area is very important, because different pharmaceutical companies, as pharmaceutical companies, they are mostly commercial enterprises, and looking at the complexity of the whole drug discovery process, it is obvious for pharmaceutical companies to have their own strategies. And most of the times, big pharma companies, they obviously work on first world, what is called as first world countries, diseases affected to the first world countries, such as cancer, autoimmune diseases, inflammatory diseases, metabolic, cardiovascular. These are the kind of areas most of the big pharma MNCs work with. However, there are several companies which also work on niche areas and orphan disease areas, and still they can make a big impact on the industry and in the drug discovery research. So unmet medical need becomes obviously the flag post for developing, for choosing a particular therapy area. And but other than high unmet medical need, various in an industrial sense, various other factors such as scientific relevance, resources, availability of resources, and various strategic market considerations are also important for companies to choose which particular therapy area they have to work. In the next slide, identifying a drug target. So at this point of time, having identified one particular therapy area, and then in that particular therapy area, what drug target we need to work with. So that also is possible. In a general sense, a drug target is normally defined any a biological entity that is associated with either a physiological process or a pathological disease state, and which interacts with and whose activity can be modulated by drug substance. So that And most of the times, as I already mentioned, drug targets most of the time comes identification of drug target comes from a basic and fundamental, lot of basic and fundamental research. Next slide, please. So let us look at some of the ideal characteristics of a good or ideal biological drug target. A drug target needs to have, first of all, it needs to have a safe profile. That means when we say safe profile, the target, there should not be any target-based severe adverse events or side effects associated with the drug target itself. Then the second, one more important thing is a confirmed beneficial role, which is verifiable at a preclinical stage. Which is verifiable at a preclinical stage itself in the form of knockout studies or knock-in studies and phenotypic studies. The third thing is, it is always desirable to have a drug target whose tissue expression is selective in nature. Even though it is not always possible, but it is a desirable thing. And having a good, perfect, protein three-dimensional structure, understanding of three-dimensional structure would be an additional thing to assess the drugability. And most important thing is, just because a particular therapeutic drug target is there, that doesn't automatically make it a good. So the one important thing to make it a good workable target is availability of various assay systems, and where availability of favorable assays, which can be a biochemical in nature or even cell-based assays, and which are amenable for high throughput screening. These are some of the important things which are which define how workable a particular drug target is. And then therapeutic index is something that the drug target should be able to provide a good window between efficacious dose and a toxic dose, which is very important for further development. And obviously, last but not least, is a favorable IP status, intellectual property status. That means free to operate. The company should be able to free to operate in a particular intellectual arena, and there should not be any blocking from other companies or any other academic researchers. Can I go to the next slide, please? Hello? Hello, am I audible?
Yes, sir.
Yeah. In the slide, having selected the target, having done the target selection, as I already highlighted, target selection is based on a complex balance of scientific, medical, and strategic marketing and business considerations need to be kept in mind. And then
సార్ చేసినప్పుడు సార్ స్లైడ్స్ చూపించగలరు, సార్ చేసినప్పుడు మనం చేయలేము. ఇక్కడ మేము చేయలేకపోతున్నాం, అక్కడేమో సార్ చేయలేకపోతున్నాం. ఇప్పుడు సార్ని స్టాప్ చేసేమనండి, మేము ఇక్కడ నుంచి యాక్సెస్ ఇస్తాం. ఓకే కదా? హలో మేడం,
Am I audible now? Hello? Am
Yes,
I?
sir. Yes, sir.
Yeah.
Sir, we'll share the screen, sir. Just wait for a minute.
Sorry, yeah.
Yeah. If both are sharing there, it is getting disconnected, sir. We'll share the screen.
Yeah, sure. So I am on slide number 12. Can you go to slide number 12?
Sure, sir.
Yeah. So having a selected target, it is important to have a validated target to start with in the initial stages of drug discovery program itself. One can say that ultimate validation for a drug discovery for a drug target comes only from the phase 2 clinical stage, but because of the complexity of the drug discovery process, it is important to have various levels of preclinical validation of the target so that we won't have any surprises at the clinical stage of the development. So slide number 13, next one. So here we can see various classes of pharmacological classes of drug targets that are as provided by human genome, and which are as you can see in the slide on the left side of the screen, proportion of various human protein drug targets that are across from major families. And on the right side, you see the small molecule drugs that target these major areas, and proportion of the drugs that are already available, which are targeting these drug targets classes. So ultimately, what is a good druggable target in the sense as we have seen, a molecular target can be a druggable. A good druggable target is a macromolecule with activity which is linked to human disease pathophysiology and amenable for modulation by a drug substance. So coming to the discovery stage, as we have seen, after having selected the drug target and had a validation, initial validation to make sure that it really works for the intended pharmacological use through various mechanistic studies and efficacy studies. So initial validations. So in the discovery stage, pharmacologists and biologists will be mainly concerned with developing various assay systems, be it biochemical or cell-based assay systems and various other animal model systems. And then medicinal chemists mainly contribute the compounds from various pharmacophores. They may adapt different particular methodologies. It can be like a huge compound library screening assisted with high throughput screening strategies support by biologists and pharmacophore analoging, structure-based design with the help of three-dimensional structure and any source of information on a chemical compound source from any published literature, or sometimes even the initial hit molecule may even come from a natural product which is available, which is known to be interacting with the drug target of our interest. So next slide, please. In the lead screening stage, in vitro studies, one particular, why there is a need for in vitro studies. So here, when we are synthesizing tens and thousands of compounds, it is not possible, it is impossible to test every particular drug moiety in vivo systems like animal studies or something. So for that, it is very important to have a very perfect fast to screen the compounds that are being supplied by medicinal chemists for their affinity and selectivity and activity. And and then once we have good pharmacophore chemical substances which are having good affinity with the target of our interest and activity and also selectivity profile, so then then the next stage of in vitro studies will come from pharmacokinetics and DMPK related various assays to ensure whether the chemical substances that are metabolically stable or not, whether they have good permeability to cross cell barrier or not, whether we can foresee any issues with drug-to-drug interactions. These certain of these things have to be analyzed at the very early stage of the early stages of the pharmacophore building and chemical scaffolds which we are working with. Next slide, please. So with these procedures, with a concerted effort from medicinal chemists and biologists, what we need to having identified a hit molecule. So then, based on the various screening. The idea is to take the hit molecule into a lead stage and lead molecule and the lead developmental stage. Next slide. So what happens in a lead optimization? From a chemistry perspective, medicinal chemistry perspective, they make use of structure activity relationship to optimize the lead molecule, to identify pharmacophores, and sometimes one has to work with several pharmacophores, because it is not okay to work with only one chemical scaffold and one particular pharmacophore, because we never know when a particular whole scaffold or a whole pharmacophore may actually fail. It can fail at any stage of the drug discovery and developmental process. So, next slide. So once we have a couple of good pharmacophores and chemical scaffolds, the next level of importance is medicinal chemists and pharmacobiologists need to work on optimizing what is called as more of a druggable properties of a chemical scaffold. So this is called as a lead optimization from a pharmacology and pharmacokinetic perspective, which is very important because until unless we don't have certain required pharmacokinetic properties in a particular molecule or scaffold or a pharmacophore, it is very difficult to have to achieve. So in industry, normally people say there are certain abilities that are required in the chemical substance. They say, first of all, a solubility, then permeability, based on permeability, bioavailability, and metabolic stability. All these things are very important in optimizing the lead to have really a druggable lead candidate. And pharmacokinetic and pharmacodynamic modeling is very important here because ultimately the pharmacodynamic effect is driven by how much dose. Hello? Hello? Am I audible?
Yes, sir. Please continue.
Yeah. So it is important to have what particular dose to have in order to achieve a good plasma concentration so that we can have a desirable pharmacological effect with a clear-cut window over safety side effects or therapeutic to achieve therapeutic efficacy. Next slide, please. So here I would like to highlight one particular thing with respect to drug discovery, especially from a medicinal chemist perspective. They normally use a particular thing called Lipinski rule of 5. So which help them in deciding what kind of particular physicochemical properties that are required in a particular chemical scaffold and a pharmacophore in order to achieve what is called as a good solubility, metabolic stability, permeability, and bioavailability. So this greatly helps. The Lipinski rule of 5 greatly helps them in achieving thing. So far here, next slide, please. So far we understood what is importance of choosing a right therapy area, and then right therapy area and right kind of drug target to work with, and doing initial validations at a preclinical level itself to make sure that there won't be any surprises in clinical development. And then in the discovery process, how chemists and medicinal chemists and biologists and pharmacologists and various other experts have to work in coordination to have a fully developed lead molecule. So once we have arrived at a lead molecule in a particular program, then extensive studies will be taken up in the form of various pharmacological studies for efficacy purpose, for understanding the mechanistic profile of how the mechanism of the molecule is working, and what kind of to understand drug-drug efficacy interactions, what is the metabolic profile, and then extensive studies. Next slide, please. Also, activities need to see safety pharmacology aspects. And the safety pharmacology issues. So all these things will greatly determine so all these things will finally make the program to go ahead. and optimizations, this thing, and there can be surprises. So the thing is, even at this particular stage, you cannot afford with only one particular lead molecule. Normally, different companies will have different strategies, and most of the times they will have different lead molecules. Sometimes probably from different pharmacophores, different chemical scaffolds to work with. And then finally, after conducting extensive studies of pharma efficacy and safety studies, ultimately a clinical, preclinical or clinical lead molecule will be selected to take it into clinical stage of development. So in the next slide, I think it is animated, and one need to click from your side one need to click several times to have a fully emerged slide. So broadly the developmental stage from preclinical to clinical can have, as we all as already seen, in vitro biology, pharmacology, and then various optimizations at the level of drug metabolism and the pharmacokinetics level. All these things, finally we select a clinical lead molecule, and then lot of role by pharmaceutics people where a pre-formulation need to be developed, and a formulation, and what kind of formulation we want to develop, that need to be decided. And then a different kind of chemists, organic chemists, where process chemistry need to be optimized, and then so that we have a constant supply of enough drug material, so that the formulators can work with on formulation and to have a formulated product to be tested in a clinical activity. Next slide, please. Next slide is also animated slide, where one need to keep on clicking to see the several stages of clinical trials. As we can see, most of the times phase 1 clinical trial is something like where it is most of the time it is done on healthy volunteers. So there the idea is to have to assess the safety and pharmacokinetic nature in a clinical. For first time we are doing it in a clinical stage. So far in a drug discovery program, we have seen that in a preclinical stage we have a very good understanding, but phase 1 clinical trial is the stage where first time the program will be tested in a clinical stage on healthy volunteers to assure the safety of the drug candidate in the human population, and also to have a good assessment of uh, pharmacokinetic nature of the molecule in humans. So then the things will get proceeded into phase 2 clinical trials, where several hundreds of patients in multi-centered trial will be tested with a randomized. Most of the times phase 2 studies are done in a randomized level, with a control group and a treatment group properly this thing. And the outcome of the phase 2 study is where we actually get for the first time clinical proof of concept, what is called as clinical proof of concept. Till now, nobody is sure whether, even though we have several stages of preclinical validation, in a phase 2 trial, first time we will get to have the proof of concept, what is called as proof of concept. And so based on a good proof of concept and efficacy parameters being achieved in phase 2 trial, the program will further go into phase 3 multi-centered trial, which will have several hundreds and thousands of patients across different continents and various ethnic backgrounds, to ensure that that in a larger trial, to have the drug product to have an excellent efficacy and a safety profile, which further goes for approval purpose. Next slide, please. So once clinical trials are done, then the the huge amount of data that is being generated here. This slide is also animated slide. I'll ask. I need assistance so that somebody can keep on clicking till the whole slide gets emerged. So the amount of all the clinical data need to be collated and properly analyzed, and then the regulatory submission here. The lot of people with the regulatory department and medico marketing people need to come into the stage, and they need to prepare what is called as drug dossiers, and investigational new drug application need to be filed to the regulatory authorities. Then regulatory authorities will set up advisory committees and regulatory and review committees. So finally give based on the data availability and whether it is confirming whether the program is having good beneficial efficacy versus safety windows. All the things taken into consideration, finally drug authorities will approve the drug product. Next slide, please. Hello? Hello?
Yes, sir. Yes, sir. So can you?
Am I audible?
Yes, you are audible.
Yeah. In the next slide, there is what is called as, see, for a successful discovery program, different companies will have different strategies. So here I would like to mention one particular strategy. Actually, this came in a publication in a drug discovery, Nature Reviews Drug Discovery. So this particular strategy is being followed by AstraZeneca. It is called as a 5 right framework strategy. So this strategy tells that for a successful drug discovery program, one need to have a right kind of a target in the first place. So that is called right target, and then right tissue, and in the sense which particular right tissue, and then right kind of safety of the target program, drug discovery program. And importance is the right kind of patients to choose. Many of the times in a clinical trial, if we don't choose the right kind of patient population, still the required clinical outcome may not be achievable. So that it is also important to have right kind of patient population, and then obviously the right commercial potential. See, for a particular drug discovery program and a particular mechanism, there may be a chance that a particular drug target and a drug substance may have different uses, therapy uses, but it is also important, to which particular right commercial potential need to be tapped, and on a risk averse in a risk aversive basis. That is also something which is very important. Next slide, please. In this slide, I would like to highlight, for the benefit of students and all participants, some of the important things in a successful drug discovery program. This I would like to call it as the 5 P's and one most important thing in drug discovery program. So the drug discovery research teams, we need to have a very passionate teams. It is immaterial how big or how small a team is, but everybody in the team, drug discovery research team, need to be passionate about drug discovery research. And in general, companies need to have a perseverance, and companies need to have a perfection and right kind of balance of scientific teams. And especially one need to be the people who are taking the decision making. The decision making management, the team management need to be practical. And the most important thing is one need to take decisions on an impassionate basis. it is often seen that across different teams we get emotional about get attached to the program and at every stage. This is something this impassionate decision making is a very important aspect in the entire stages in all the stages of drug development. The idea is to always push forward the right kind of ideal candidate or the best possible candidate to the next level. Because this is very important, because the whole drug discovery process is so complicated and so much of risk. There is so much of risk involved, risk of time and risk of resources. All these things, keeping in that matter, keeping all those things in mind, one need to have a impassionate decision making decision making management authority. So these are certain things which entail a successful drug discovery program. Next slide, please. So obviously, focus and commitment are very important for a drug discovery program. Thank you very much, and I would like to now the session is open. If any questions are I welcome the questions from participants. Hello.
Hello. Hello.
Sir, is it audible to you?
Yeah.
Yeah, very much. Sorry, there is some connectivity problems or something. I don't know. Maybe it is raining very heavily in Mumbai. Maybe there may be some, by Wi-Fi and all. I think there may be some issues. But I suppose the presentation went well. You need to tell me because I am not able to see half of the time. I am not able to see slides on the Teams platform. I am not able to access my slides. I had to access from my computer here, laptop.
Yes, sir.
Yeah. Hello.
Sir, Dr. Rajendra Prasad is asking you a question, sir. Sir, please.
Yeah. Hello. Good morning, sir. This is Rajendra Prasad.
Yeah.
Yeah. It's a very nice presentation with a very good specific examples. Okay.
Yeah.
And I have a question, sir. Actually, if you target if you see the pandemic disease like antiviral disease, so what is the role of organic molecules or the role of natural products? Because we require we need to target the disease in a usually the lower time, because we can't wait for the longer duration in the drug discovery process. So what is a very good strategy for to complete the process in a rapid way?
Yeah, so the thing is, my topic is restricted to a broad overview. My idea is to give a broader outer view of the drug discovery program that is in a conventional method. So to answer your specific question, in a pandemic situation, right now we are all in a corona kind of a pandemic situation where we cannot wait. So in that particular aspect, what need to be done? So that is something called as, there is a particular thing called as what is called as a drug repurposing. That means drug repurposing is something where the drug repurposing strategy, you already have so many drugs out there. So basically, they may be allopathic drugs, or sometimes they may be Ayurvedic drugs, or some other herbal things which have a potential or something. But most of the time, it would be easy for, if you have a already approved drug, whether we can, based on the particular pathophysiology of the disease. Here in this case, corona. Corona is a pandemic disease. Now we know certain things. There is lot of scientific information is known about the coronavirus and the SARS-2 novel corona COVID-19 virus. So what happens is, basically based on the current available scientific information, we need to need to look into already available drugs which are already present for some other disease purpose or some other indication. And we need to quickly take a look, take a review and test. So just for example, I will tell you how this drug in a particular pandemic situation like this, how several healthcare authorities and companies and several others, healthcare workers, clinicians, have quickly come up with various, treatment modalities to test with and to test and see whether they are effective or not. So if we see the very first indication, everybody in combination. So this is, we all need to keep in one thing in mind, that COVID mainly affects the respiratory system. So that is one of the one particular aspect, and it is already known in scientific literature that hydroxychloroquine, it has a very peculiar mechanism of action. If ultimately it has a role in viral replication. Apart from even though it is used in malarial parasite and killing the malarial parasites, like the anti-malarial drug also has a good antiviral properties, where the replication of the viral replication in a can be inhibited with hydroxychloroquine, and this information is known. So based on that, they quickly tested this particular thing, and what is known in literature, that hydroxychloroquine is an ionophore. At a very molecular level, it works as an ionophore. It allows entry of zinc into the cell, and we all know that in a molecular, from a molecular biology perspective, there is already prior information that zinc has a role to play in optimizing the viral replication. So when we have a particular virus which is entering into the cell and proliferating at a very high level, so people thought that it would be a good strategy, and initially there is some level of success. But though hydroxychloroquine is a lot, I'll say, like it has its own efficacy, but it has so many issues, especially with respect to cardiovascular system. It has what is called as a side effect called QT prolongation. So in a ECG, we know there is a QT curve, PQRT curve. So the QT prolongation, which is a very debilitating side effect for hydroxychloroquine kind of compounds. So it may not be safe. That's why ICMR and WHO and all, even though they initially given extensively hydroxychloroquine to every other patient, but the doctors immediately realized that it is people who are already fragile, who have already very severe form of disease. For them, people may actually die because of cardiovascular side effects. So then they came up with a different strategy, that rather than patients, the healthcare workers, the frontline healthcare workers, doctors, nurses, they can be treated with this particular drug, and most of the time they are very healthy. So that those people who are at the frontline health workers, who are continuously working with patients who are having this infection, so that those people don't fall sick. Yeah, I suppose I have answered you. The same applies for Ayurvedic products as well.
Yes. Yeah. Thank you, sir.
Yeah.
Sir,
Yes.
sir, I really like the concept of AstraZeneca 5-8 framework and 5 Ps.
Yeah. Yeah, because from the slide one, I tried to there is a particular purpose why it is very important to highlight a lot of people, especially I know you people from an academic background. You may not be knowing so many nitty-gritty details of how difficult it is, why it takes anywhere between 10 to 12 years to develop a drug from lab stage to clinical stage. And I tried to highlight various expertise that are required and various stages of discovery program, and at every stage there is a risk. That is also one of the reason. We are all pharma graduates. We need to make an understanding when companies are doing, not everybody comes forward to do this kind of risk investment. And there is in a drug discovery program is it is a very high risky programs. And obviously it is sometimes it looks obvious for companies to have their own commercial thinking. Because otherwise it is impossible to sustain so much of risk investment, and at the end of the day not achieving anything in the form of an approved product. So keep in mind, we need to have checkpoints and validations and revalidation. It can be a preclinical validation, target level validation in animal studies, and then so many aspects need to be kept in mind to finally arrive at a particular drug program. As I explained in one of the slides, in order to achieve one approved drug, after 12 years of research and heavy investment, risk investment, you need to start with, you need to develop, you need to synthesize at least, from a medicinal chemistry perspective, one may need to synthesize several hundreds and thousands of compounds, and then meticulously screen them for their affinity towards the target of interest and the potency and so many other things. And then comes the role that, once we take a drug, it need to be bioavailable, and it need to achieve the required drug concentrations in the particular tissue where it is supposed to work. All these things need to be kept in mind. So every company has their own strategy. Most of the times they don't divulge this thing, but this is like AstraZeneca is good enough to publish their particular strategy. Which I am sure more or less other major pharma companies are also more or less adapting similar kind of strategy, even though they don't want to disclose for secretive purposes.
Yes, sir. Thank you very much, sir. Like, any questions?
Yeah, one thing I would like to highlight, I tried especially students participating and also faculty who are participating in this webinar series. There is, for pharma students are at the right place. As I said, in the pharma industry, there are several particular aspects of pharma industry, and we can have right from people from pharmaceutics, pharmacology, pharmaceutical analysis, and even other specializations, biotechnology or some others. Every student, there is a role, there is a meticulous role and role to play for different people from different specializations, and formulation, especially pre-formulation. Pharmaceutics and pharmacokinetics. Pharmaceutics people can come into pre-formulation stage and formulation stage, which also happens in drug discovery research. In a generic research, anyhow, they have a huge use. The application of formulation scientists are very much required in generic, but also in discovery research programs, their utility is very high. And same is the case with pharmaceutical analysis, bioanalysis. In the entire drug discovery and pharmacokinetics and drug metabolism, there is a very great role for bioanalysis, and that is they can play a major role in those things. And obviously, I highlighted the medicinal chemists and pharmacologists and pharmacokinetic experts in pharmacokinetics. Their role is more or less goes, and then till the end of preclinical stage, all these experts are required. And after that, you need different kind of expertise, especially clinical pharmacologists and clinicians and regulatory and other marketing and medico marketing and legal, so many other experts. So there is a good amount of for students, they have to realize and understand way and based on their interest, ultimately what they really want to. So these are certain options I try to highlight in the presentation so that they can choose.
Yeah, thank you very much, sir, for taking your time and giving your most valuable information on drug discovery. Actually, these words we didn't know anything about this. These are the industrial expertise. So you are enlightening our knowledge at the academic level, sir. And I would like to say sorry for all the participants, like that inconvenience had occurred due to heavy rain in Mumbai. So sir has some technical problem, because of that slides was not shared properly. I'm sorry for that, and thank you very much, sir.
Yeah, I hope I have shared the slides. I think you can share those slides with your participants also. Probably they might have heard me, and then they can go through the slides and they can recollect and they can correlate all those slides. Sorry from my side, because there is a heavy rain and I think my broadband highway Wi-Fi network is also going on and off. It looks like so that's okay.
I will share your presentation.
This is the part of yeah, sure, you can share it, no problem.
Yeah, we have WhatsApp group for the participants, sir. I'll share the presentation at WhatsApp group, sir. So the participants will look into that.
No problem. You can share it. You can share it because it is like it's a broad overview perspective, and there is nothing confidential I have shared. Normally, we will have issues if I am sharing some confidential data or something. But there is nothing confidential as such, so you can very well share with all your participants, especially your students and all, for them to have, to go through the slides one more time to appreciate what we have discussed so far.
Yeah, thank you very much, sir, for your time. Yeah.
Okay.
Yeah. Have a good day.
Bye bye. Yeah, same to you.
Yeah. Thank you for all the participants. Next, we have tomorrow session starts at 10 o'clock.
Okay.